Hello everyone and welcome. My name is Violet Aswa Akek. I am a lecturer at the Department of Clinical Medicine, KMTC, Nairobi campus. Today I'm going to be talking about inflammation, and this lecture is purposely made for students of clinical medicine, but anyone who is interested can still follow it through. So welcome. A few introductory statements that I'm going to make. First is to understand what is inflammation. And so the word inflammation is taken from a Latin word inflammare. And the meaning of this word in English is to burn. What that would imply is that any time or any tissue that experiences or undergoes inflammation will usually have a burning sensation. And if you examine that particular site or area, you will feel or you will be able to pick increased local temperature. So remember the word inflammation taken from the word inflammare, which is a Latin word meaning to burn. It is a local response to living tissue, and that response is usually from any type of injury or injuries or insult caused from any source, from any etiology. Therefore, we are saying that when there is an inflammatory response, this will usually be caused by any type of injury. What is therefore the purpose of inflammation? Threefold. The first one is to localize the causative agent and also to try and eliminate it. That means, by extension, it limits tissue injury. And when tissue injury is limited, the best outcome is that that tissue or cell is restored back to its original form and function. Remember, we said pathology is a study of disease. In other words, disease, disturbances caused by disease affecting both the structure and the function. Therefore, when inflammation takes place, one of the benefits or the reasons that happens is to localize and eliminate the causative agent, to limit tissue injury, and also to make sure that the tissue goes back to its normal functionality and structure. Therefore, we are saying that inflammation is not a disease. Rather, it is a physiologic response of living tissue to injury or insult. So, commonly when an organ or a tissue is inflamed, there will be a way that it is described. And you will usually know that description by the suffix itis. So we shall give a few examples. For example, inflammation of the liver would usually be referred to as hepatitis. Now, let me say this at this point, that usually that description is picked or derived from the cell type or the tissue type that is inflamed. The liver cells are known as hepatocytes, and therefore inflammation of the liver would usually be referred to as hepatitis. If it is inflammation of the appendix, again from the word appendix, then you would say appendicitis. Now, there are few exceptions. For example, the lung. Inflammation of the lung, rather than being referred to as pneumonitis, because the cells of the lungs are called pneumocytes, or rather than saying lungitis, we say pneumonia. That is just one of the few exceptions. However, there is another condition known as pneumonitis, which is usually as a result of heavy metal deposition into the lung parenchyma. Another example of exceptions where the suffix itis is not used is inflammation of the pleura or pleural tissue. In this case, it is known as pleurisy and not pleuritis. Inflammation of the skin. The cells of the skin are known as dermatomes. So we don't say skinitis, we say dermatitis. All right? Now, back to the other example where itis works, hepatitis, appendicitis, arthritis, and so on and so forth. So remember that. Edema formation causing pressure on the blood vessels. Then you may have stasis of blood and ischemic injury. All these things will just contribute to further tissue damage. So, having said that, now let us look at the cardinal signs of inflammation. I want you to remember that a cardinal sign is usually referred to or is understood as the primary or major sign by which a diagnosis is made. It means when you see these cardinal signs, you know definitely there is an inflammatory process. What are they? One is redness, also known as rubor, heat, also known as color, pain, also known as dolor, and swelling, also known as tumor. These are the 4 main cardinal signs of inflammation. Now, there is a loss of function as well. And some books will usually list loss of function as a 5th cardinal sign of inflammation. Some will not. And the explanation is loss of function, its presence or absence does not expressly denote the diagnosis of inflammation. So, loss of function is a feature, may or may not necessarily be a cardinal sign of inflammation. Okay. Having made those introductory statements and having mentioned the cardinal signs, let's now look at the real thing that happens during the acute inflammatory reaction. Now, the changes that take place during an acute inflammatory reaction are usually threefold. The first and foremost, like we said, is the change in the vascular caliber and flow. And we shall see what we mean by that. At the same time, besides the changes in the caliber of the blood vessel and the flow of blood through it, there will be increased vascular permeability. When there is increased vascular permeability, that will aid the formation of inflammatory exudate. And at the same time, remember, like we mentioned in the introductory statements, it's not only the fluid, protein-rich fluid that will escape, but also white blood cells will be attracted to the site of injury and they will escape from the blood vessel into the tissue where they are needed. Now, like I said, these processes will usually lead to the formation of the inflammatory exudate. There will also be accumulation of fibrin at the site of injury and white blood cells. And like we said, they take place where? At the level of the microcirculation. So, let's talk a little bit about what happens at the level of the microcirculation under normal circumstances. So, like I told you, the microcirculation will usually begin at the level of the arteriole. Remember, the arteriole is at the tail end of the artery. And then it ends at the level of the venule. The venule is the beginning of the veins. The in-between the arteries and the veins is where we have the capillary bed and what we call the microcirculation. This is where exchange happens. Gaseous exchange as well as nutrition. So, usually, under normal circumstances, blood will usually enter the microcirculation via the arterioles. And because the arteriole is the tail end of the artery, it has the nature of arteries, which is usually a comparatively narrow lumen and a thick muscular wall. And then it will usually leave via the venules, which is the beginning of the veins. And again, the venules will have the characteristics of veins, which have a comparatively larger lumen and a thin or thinner muscular wall. Now, in between the arteriole and the venules are what we call the metarterioles and the capillaries. So you have the arteriole, metarterioles, capillaries, and then the venules. Now, these metarterioles are usually surrounded by smooth muscle fibers. Muscle fibers, the smooth muscle fibers have a particular characteristic, that is the ability to constrict and to relax. When they constrict, they cause the lumen to narrow, and when they relax, they cause the lumen to expand or to broaden. Therefore, because of this characteristic, that or whatever they have, the smooth muscle fibers, they are able to relax and contract. That way, they are able to control how much blood enters through the capillaries in terms of volume. Therefore, we can comfortably say that they act as pre-capillary sphincters because they regulate the flow of blood into the capillary bed. Okay? Let me just say that a sphincter is a ring of muscle surrounding and serving to guard or close an opening. So we are saying metarterioles are the precapillary sphincters. Remember I told you arterioles, metarterioles, capillaries, venules. Metarterioles are fashioned with smooth muscle fibers. They have ability to constrict and to contract. When they contract, the amount of blood going to the capillary bed is reduced. When they relax, the amount of going to the capillary bed is also increased, depending on the physiologic demand. It is usually a response to physiologic demand. That is what happens at the normal, at the microcirculation under normal circumstances. So, how does the flow happen? At the capillary bed, some capillaries are large than others. That means we have large capillaries and we have smaller, comparatively smaller capillaries. Under normal circumstances, blood will usually prefer to flow through the larger capillaries. But when there is increased physiologic demand, then even the amount of blood through the preferential channels, which is the bigger capillaries, will increase. And also, if the demand is higher still, then even the smaller capillaries will open up and start bringing in blood. That is usually following a physiologic demand. Maybe if an individual is exercising or they are running or anything like that, anything that causes increased demand of oxygen supply or nutrients. So that means that the flow through the capillary bed is intermittent. Sometimes it is high, sometimes it is low, depending on the physiologic demand. And what enables the fluctuation? It is the relaxation and contraction of the precapillary sphincters, which are also known as the metarterioles. Okay. Having said that, so what happens during acute inflammation? The inflammatory mediators are released, and it is these inflammatory mediators that will act on the blood vessel wall, remember, to alter the permeability, to alter the amount of blood flowing through, and so on and so forth. So there is usually, initially there will be transient vasoconstriction of the arterioles. There will be widespread vasodilatation. There will be what we call active hyperemia. There will be increased vascular permeability with exudate formation and stasis of blood. Number 6 would be now accumulation of white blood cells, which we shall look at a bit separately. So, following injury or of any sort, there is transient vasoconstriction of the arterioles. Note that this transient vasoconstriction does not always occur. It may not occur. However, there is widespread dilatation of the arterioles. This one occurs all the time. However, the initial transient vasoconstriction may or may not occur. And then there is widespread vasodilatation, a function of the inflammatory mediators. When this happens also, there is opening up of all the other small blood vessels. Remember I told you at the capillary bed, some capillaries are bigger than others. Under normal circumstances, blood will usually be flowing through the bigger capillaries. But when there is inflammation, all the small capillaries also open up and they begin to allow blood to flow through the capillary bed. So we have the usual large capillaries having the blood flow increased, and also we have the smaller capillaries opening up and also flowing blood through the capillary bed. That increases the local blood supply of flow to the site of injury, and that is known as active hyperemia. And usually when there is an inflammatory response and all these blood vessels are opening up, there's blood vasodilatation and all that, the amount of blood flowing to the injured tissue will usually increase by up to 10 times. Then there is increased vascular permeability, like we said, which is responsible for formation of the inflammatory exudate. Like I said before during the introduction, is that an exudate is protein rich. And usually, if you would send it for chemical analysis, you'll find that the protein level is between, then let's just say it is beyond 35 grams per liter. When it is more than 35 grams per liter, definitely the cause of that exudate or edema is inflammatory in nature. And so you should be starting to think about what could be the differentials in line of those things that can cause inflammation. If the exudate has protein level which is less than 35, that is a transudate, and therefore you should be thinking of differentials that cause non-inflammatory edema. That is important for you as a clinician. So, the formation of this exudate is usually because of leakage of fluid from the intravascular space, which is accompanied by escape of proteins. And usually, when that would happen under normal circumstances, any excess would be drained by the lymphatics. So let us see. What influences the amount of exudate, the amount of edema? The main thing is how much space is available. That means inflammation happening or taking place in cavities, areas of the body where there are cavities like the peritoneum, then that area, a lot of exudate will form because there is a lot of space. But if it is happening in more tighter tissues like the liver and things like that, in the muscle, then less exudate will form. That is one. Okay? And also depends on how much fluid is able to pass through the capillary wall, whose permeability has increased. Okay? Now, let's look at this further, but then we shall look at what would be the situation in a normal microcirculation versus then we compare with what happens during inflammation. We've already seen that under normal circumstances, we'll have the preferential or the bigger capillaries being the principal suppliers of blood through the microcirculation. But then if there's the physiologic demand, then that blood supply is increased. What increases or decreases the flow? It is the meta-arterioles, because they have the ability to constrict and to relax, and therefore control the amount of blood flowing in. But then we are saying during inflammation, there is vasodilatation, the vascular caliber is changed, the flow is increased, so you have as much as up to more than 10 times what the capillary bed usually receives at the site of inflammation. Another thing that we need to compare is how diffusion or movement of molecules happens. So we are saying that under normal circumstances, there will be diffusion, of small molecules along concentration gradients. And this will usually include oxygen molecules, glucose, or carbon dioxide against the concentration gradients. Now, when you remember what you learned in high school in biology, when a substance is moving, or if a molecule is moving against along a concentration gradient, it means the molecules are moving from the place of high concentration to the place where the concentration is low. So at the level of the capillaries, at the microcirculation, that is at the capillary bed, oxygen will usually be moving from the blood into the tissue. Why? Because it is of higher concentration within the blood vessel in the blood because of hemoglobin, which is coming from the lungs, and so it will be releasing oxygen molecules to the tissue. And because the carbon dioxide in the tissues is higher, it will now be moving from the tissues into the blood vessel, being picked by the HB and red blood cells and being transported back to the lungs for oxygenation. However, heavier molecules, fluid and other solids, heavier molecules will usually be transferred by ultrafiltration. Fluid and other heavier molecules or solids will usually be transferred by ultrafiltration. Now, this ultrafiltration is controlled by the balance between what we call the hydrostatic and the colloid osmotic pressure, also referred to as oncotic pressure. Hydrostatic pressure is exerted by the presence of water molecules, and the colloid or oncotic pressure is exerted by the presence of protein. Hydrostatic and colloid osmotic or oncotic pressure within the blood vessel. Hydrostatic, water; colloid osmotic or oncotic, protein. This balance is what makes sure that you don't get edematous. So normally, the hydrostatic pressure at the level of the arterial or at the beginning of the capillary bed is higher than that of the protein or plasma oncotic pressure. And at that point, because the hydrostatic pressure is high, then there will be flow of water from the blood vessel into the tissue at the capillary bed. Blood is now continuing to flow through the capillary bed. And so what's happening? As it is continuing to flow, the hydrostatic pressure, remember, is higher at the beginning of the capillary bed. And so water is leaving the blood vessel wall by ultrafiltration into the tissue space. But as the flow continues towards the end, the hydrostatic pressure is reducing gradually because water is leaving. By the time this blood is reaching towards the end of the capillary bed, the oncotic pressure is now higher than the hydrostatic pressures. That then changes or reverses the process. So water now again is reabsorbed back into the circulation, back into the blood vessel wall. At the end of the day, there is no edema because what was extravasated has been reabsorbed, and any little excesses are removed by the lymphatic system. So under normal circumstances, that is what happens. At the beginning of the capillary bed, hydrostatic pressures are higher than oncotic pressures. That will allow water to extravasate by ultrafiltration through the blood vessel wall, whose permeability has anyway increased. And then, under normal circumstances, the blood vessel wall is normal. So the hydrostatic pressure will pull or release water outside the blood vessel wall. But towards the end, Comparatively, the hydrostatic pressure will be lower than the plasma oncotic pressure, and that will reabsorb water back into the blood vessel, and therefore there is no accumulation of fluid in the tissue, no edema. However, during the process of acute inflammation, the dilatation of the blood vessel wall will usually result in intravascular hydrostatic pressures that are high, normally at the beginning of the capillary bed. Remember, we said that the blood flow to the site of inflammation is increased by up to 10 times. So, while the blood is flowing through the capillary bed, yes, the hydrostatic pressure will be reducing along the gradient, but by the time it is getting to the venule, still the hydrostatic pressure, while it has reduced, it still exceeds the oncotic pressure. And because it exceeds the oncotic pressure, there is no reabsorption of this water back into the blood vessel wall. And that accounts for the development of edema. And because the intravascular spaces of the blood vessel wall are now, the interspace between the endothelium is increased because of the increased permeability, protein will also be escaping together with the fluid. And so the hydrostatic pressures will still be high. There will be no reabsorption of water back into the circulation, which means now it has accumulated, and we call that edema. Not only will there be edema, but also protein will have escaped. Lots of protein molecules will have escaped through the blood vessel wall into that edematous fluid or exudate, and that is what we call as exudate because of the increased protein level. I hope that is clear. So, if the inflammatory reaction is very severe, large amounts of exudate will form, large amounts of protein will also escape. Okay? And how does that happen? Remember, we talked of the inflammatory mediators. Some of them are vasoactive. They are the ones which act on the endothelial cells, causing them to shrink. And when they shrink, the interspace between those cells is increased. That is how the permeability is increased, allowing all this escape into the tissue space, which we also call the third space. Now, we also talked about slowing of blood at the point of inflammation. Now, these are the reasons why you will have stasis or slowing of blood at the point or the site of inflammation. Number one is hemoconcentration. Hemoconcentration just means there is less water in the blood vessel wall, within the blood vessel cavity, with lots of solutes. The blood cells are there, and so against a reduced or contracted amount of blood, that causes hemoconcentration. When there is hemoconcentration, there will be increased viscosity. When there is increased viscosity, the flow of blood will be slowed, and therefore there will be stasis. Now, as the edema also continues to form, it has pressure effects on the surrounding blood vessels. Again, because of this pressure, then on the blood vessels, that will tend to slow the blood flow, stasis. The third reason is narrowing of the blood vessel lumen. Why would the blood vessel lumen get narrowed? This happens when the white blood cells are trying now to move out from the blood to the tissue where there is injury. So initially they will line themselves along the endothelium. While that happens, while they align themselves along the endothelium, that will contribute to narrowing of the vascular lumen, before they extravasate into the tissue. So these are the three main reasons why you will have slowing or stasis of blood. Increased viscosity because of increased hemoconcentration. Why? Because there's lots of fluid leaving the blood vessel into the tissue, but leaving behind the cellular elements. 2, the pressure that is exerted by the swelling caused by the inflammatory exudate, and also narrowing of the blood vessel lumen caused by margination or the lining of or aligning of the white blood cells along the endothelium while they await to move out into the tissue. So let's look into a little bit detail on how the white blood cells are attracted. They leave the blood vessel wall, they leave the blood through the blood vessel wall to the site of injury, and what they do. So firstly, it's important to remember that it's very important that white blood cells are at the site of the injury or inflammation, because the inflammation could be caused by a microorganism, and their presence is important because then they will begin to fight the microorganism. Secondly, the tissue injury may be associated with death of cells, and these cells need to be removed by the process which we call phagocytosis, and we shall mention a little bit later. So among the white blood cells, there are different types, subgroups. We have neutrophils, we have lymphocytes, we have monocytes, basophils, and so on and so forth. So among these white blood cell groups, the first white blood cell type that appears at the site of inflammation are the neutrophils. They are also known as polymorphonuclear leukocytes. This will usually appear as the first white blood cells, whether it's due to the injury is due to an infection or not, whether there are dead tissues there or not, white neutrophils will always show up first within the first 24 hours. Now, as time goes by, maybe the cause of inflammation is infectious, and maybe the infection is due to bacteria or virus or fungus or something else. After the first 24 hours, then a specific type of white blood cell will be predominantly attracted to the site of injury, depending on which type of white blood cells is predominantly attracted to come and fight. And remember, these white blood cells are attracted by what we call chemotaxins. They're inflammatory mediators. Okay? So neutrophils will be the first to appear. Their prolonged presence or continued attraction will usually mean something else. So monocytes will usually be predominant in the later stages of inflammation, but initially it will be neutrophils and then later others, including monocytes. So the appearance of white blood cells will usually involve 4 stages. The first is margination, what I said, lining or aligning themselves along the endothelium. Then secondly, they will now move out of the blood vessel through the blood vessel wall to the site of injury by active amoeboid movement. And then there will be the chemotaxin effect. Those chemicals that are mediators of inflammation, and some of them are actually produced by the disintegrating microorganisms, the microorganisms themselves also. So they are then attracted to that site, and then once they are attracted, they have to be activated. They have to be activated so that they can act. Okay? So we said the first 24 hours expect neutrophils, later expect monocytes. But, like I told you, there could be other dominating subtypes or subgroups of white blood cells depending on what is causing the infection, if it is an infectious cause of inflammation. So, if they are bacteria, if it is a bacterial cause of inflammation, then the neutrophil attraction and pooling will be sustained. That means if you do a full blood count and ask for differential white blood cell count, and you find in your interpretation of results that it is predominantly neutrophils, that should suggest to you that the cause of inflammation is an infection, first of all, and number two, it is a bacterial infection. If the predominant white blood cell subgroup is lymphocytes, that means, yes, the cause of inflammation is an infection, and that infection is viral. If it is parasitic infestation, then the predominant white blood cell type will be eosinophils, and so on and so forth. So this is some of the things that when you do as a clinician, they help you to be able to now start segregating the different causes of whatever illness it may be, and then be able to know what kind of treatment or management to institute. So usually when there is inflammation, the white blood cell count will shoot. It will be increased in response, but then going forward, initially it will be the neutrophils, irrespective of the cause. But then going forward, if the neutrophil attraction is predominates, then that is bacterial infection. If it is viral infection, you'll have predominantly lymphocytes, like we have already mentioned. Lastly, I said the fourth stage is activation of the leukocytes. They have to be activated. Once they are activated, then they are able to phagocytose. Phagocytose is to kill the invading microorganisms and also to remove the dead matter or material. So we are saying that phagocytosis is the process of engulfing and internalizing of particulate matter. Particulate matter may either be the damaged cells which need to be removed, or the microorganisms which also need to be removed in order to control the infection. Remember, I told you one of the reasons or important reasons we must have an inflammatory response is to eliminate the causative agent. Now, in terms of microorganisms, that's very important. These specialized cells that cause phagocytosis are known as phagocytes. And the most common phagocytes that you will encounter are the polymorphonuclear leukocytes, at the neutrophils, monocytes, and tissue macrophages. Now, for phagocytosis to be successful, remember initially for the attraction and movement of white blood cells to be successful, it is aided by chemotaxins. Now, once they're there and they've been activated, for successful phagocytosis, there are things we know that are called opsonins. Opsonins are what help the white blood cell or the phagocytes to stick together with the bacteria or dead matter so that it can be phagocytosed. So opsonins are also very important. So the process of ensuring that adherence between the phagocyte and the matter to be phagocytosed is referred to as opsonization, and the chemical factors involved are known as opsonins. These are the three major opsonins in the blood. Certain antibodies of the IgG group or class, and we shall talk about IgG later when we talk about the immune response. Activated complement system, this we shall discuss in detail when we are talking about mediators of inflammation. And then we have the plasma fibronectin. Now, once attached to the cell membrane of the phagocyte, the particulate matter to be engulfed is taken in by pseudopodia and closed into a membrane known as the vesicle or phagocytic vacuole or phagosome, and then it is disintegrated and removed. That is how the body gets rid of invading bacteria. That is how the body gets rid of dead tissue that is no longer needed. So I think up to there we shall take a break and then we shall continue.